con las presentaciones de hoy. Es buenísimo estar aquí en este último día de curso, con un curso que fue realmente muy bien aprovechado por todos que están aquí. Sentimos como si todos se sintieran bienvenidos realmente, hablando su idioma, tranquilos, y pudimos hacer un curso que realmente fluyó muy bien. A pesar de tenermos portugués, espanhol e inglês todos conectados, creio que foi super bem e pudimos manejar realmente de uma maneira espetacular, que não creíamos que al princípio seria assim, e foi realmente genial. Então, empiezo apresentando a la doutora Paola Casanello, que é professora titular do Departamento de Obstetrícia e do Departamento de Neonatologia da Escola de Medicina em la Pontificia Universidad Católica de Chile, em Santiago de Chile. Dirige el Laboratorio de Programación Perinatal de la Facultad de Medicina de la PUC desde 2010 y su línea de investigación es interdisciplinaria y se centra en el estudio de la nutrición materna, el crecimiento y la adiposidad fetal, la inmunidad neonatal y los mecanismos que conducen a enfermedades crónicas en los hijos de mujeres con obesidad. Forma parte del Consejo Internacional de la Sociedad Internacional DOHaD. Muy bienvenida, Paola está aquí con nosotros desde el principio como coordinadora del curso también y estamos muy felices por estar aquí hoy coordinando esta última sesión. La doctora Débora Sabatelli es pediatra y neonatóloga graduada de la Universidad de Buenos Aires, Argentina, y actualmente se desempeña como jefa de sesión de la Unidad de Neonatología del Hospital Fernández en Argentina. Además, es la presidenta del Subcomité DOHaD de la Sociedad Argentina de Pediatría. Un placer también recibirte aquí hoy, Débora. Y ahora, por favor, la coordinación está con ustedes.
Bueno, muchas gracias por invitarme a esta tarde de aprendizaje. Bueno, a mí me enviaron la presentación de Patrícia en inglés, pero bueno, voy a tratar de traducirla al español porque me siento más cómoda. Bueno, Patrícia Pelufo Silveira es una profesora asociada del Departamento de Psiquiatría de la Universidad de McGill y directora científica de Genómica y Epigenética del Pillar of the Landmark Center para Neuroinformática y Salud Mental. Esto tiene localidad en el Douglas Research Center. Tiene amplio entrenamiento multidisciplinario en pediatría, seguimiento de neonatología y neurodesarrollo y neurociencia. La doctora Silveira también publicó más de 180 trabajos de revisión de pares y sus trabajos fueron citados más de 3.200 veces. Y en 2022 ella fue electa como miembro del colegio del Royal Society of Canada y actualmente es Senior Fellow en el Centro de Desarrollo para los Niños en la Universidad de Harvard. Muchas gracias por compartir esta tarde, Patrícia.
Muchas gracias, Débora. Gracias por la invitación y por todos que están acá. Compartilho minha tela, Cris.
Isso, ali embaixo tem um quadradinho com uma flechinha. Acho que apresentar a tela inteira geralmente funciona melhor.
Tá. Tá me pedindo para aceitar aqui, só um pouquinho, para poder gravar.
Qualquer coisa, Paty, se tiver algum bloqueio, pode me encaminhar a apresentação por e-mail, que eu abro ela por aqui.
Eu acho que vai ser mais fácil, Cris.
Tá.
Porque ele está me pedindo para... Ele está me pedindo... Tá, ele está me pedindo para reinicializar e entrar de novo.
Tá, melhor então assim.
Peraí.
Quizás para rellenar, mientras Patrícia puede enviar su presentación a Cristiane y que la pueda compartir, decirles que Patricia está dentro prácticamente del grupo fundador de DOHaD en Latinoamérica, ¿no? Desde sus comienzos en el primer encuentro que hicimos en Ponta Grossa, organizado por Paulo Matías, donde tuvimos la suerte también de conocernos con Patricia. Estaba trabajando en ese tiempo en la universidad en Porto Alegre y ejerciendo como neurocientífica, más bien en ese tiempo en modelos animales, ¿no, Patricia? Esencialmente en ratones y haciendo intervenciones, pero también había hecho pasantías en la Universidad de McGill con el profesor Michael Mini, con quien tuvo un periodo también de formación postdoctoral, creo recordar, ¿cierto? Y entonces, desde ese tiempo, Patricia ha sido muy importante como pilar desde Brasil apoyando el desarrollo de DOHaD. Así que también importante destacar eso para aquellos que no la conocen. Éramos muy jóvenes, Patricia, pero parece que ya no somos tan jóvenes. Entonces hay mucha gente que puede no conocer nuestra generación de investigadores. Mucha gente joven, sobre todo en este curso.
Muchas
Sí,
gracias, Paola.
Ya nos salieron canas, ya tenemos algunas canas, ¿sí? Bueno, Patricia ahora es una muy destacada profesora, ha hecho una carrera realmente brillante y esperamos tenerla también en nuestro congreso en DOHaD en Buenos Aires el próximo año, porque hemos propuesto ahí que así sea, mientras armamos la parrilla de investigadores, el comité científico ha propuesto tu nombre, así que ojalá que te veamos allí también, con los investigadores de tu grupo.
Muchísimas gracias, Paola.
Ahora ya está bien, excelente.
Entonces, más una vez, muchísimas gracias por la invitación. Estoy muy, muy contenta de estar acá. I'm going to speak in English because I think it's going to be easier than me trying to speak my poor Spanish there. But yeah, Chris, do you mind going to the next one, please? So I'm going to talk to you today. The proposed theme is translational studies in DOHaD, and my lab has been focusing on translational work. A lot is due to the training that I had. I was fortunate to be trained in medicine and in pediatrics, and at the same time had the experience to work in a basic science lab, working with animal models at UFRGS in Brazil, and this, you know, was such a fundamental part of my training from the beginning that it made me think of science always in a translational way. So I know this is not true for everyone, so I'm really grateful for the opportunity to have being able to be trained like that. And my lab focused a lot on prenatal stress and prenatal adversities and how that affects the trajectory for growth, neurodevelopment, and health and disease in the long term. From a clinical perspective, I was always very interested in fetal growth and low birth weight. So we focused a lot on this feature and how that would influence health and disease. So of course, the DOHaD, especially the initial studies from DOHaD, were very dear to me and very inspiring from the beginning. Now, of course, this happens in mid-80s there. And I was so happy to see this special issue that was brought up by The Lancet last year. This is a whole special call for attention to low birth weight babies that was published last year. And in this issue, they proposed this concept of a small, vulnerable newborn. What it means is that if you put together children that are born small for gestational age, preterm, or low birth weight, meaning below 2.5 kilos, 25% of the population worldwide will be classified as a small, vulnerable infant. Imagine 25% is 1/4 of the population in the world is born affected by one of these three situations. And this cuts across any sort of socioeconomic status, hemisphere, political side. It's really consistently affecting all kinds of populations. There are many reasons that can lead a baby to not grow as much as they should in uterus. It goes from the classical malnutrition, but also other things like hypertension, type 2 diabetes, or gestational diabetes, even though some of them are aligned with high birth weight or excessive fetal growth. But part of these cases are also associated with small for gestational age babies. And all situations that lead to placental insufficiency, chronic maternal diseases, stress, inflammation. As we know in this group here that are interested in DOHaD, these babies are an increased risk for developing type 2 diabetes, hypertension, metabolic syndrome as adults, but also psychopathology. So they have an increased risk for any kind of psychopathology that you can think of. There are studies showing that they have increased risk. I'll name a few, but it's really any kind of mental health problems. So schizophrenia, addiction, strongly associated with substance use disorders, anxiety, depression. So, of course, from an epidemiological and public health perspective, this is super important. It's estimated that between 25 to 60% of chronic adult disease are associated with early life events and features. So it's very important for us to understand these relationships, and all of us here share this common interest on this. You can go next, Chris, please. And my lab has spent a lot of time investigating these babies that are born small. Particularly, we were the first ones to describe that these kids have a specific way to behave towards food. They prefer to eat more palatable foods as opposed to healthy, nutritious foods. If they have the chance to choose, they're going to go for the high fat, high sugar foods as opposed to healthy options. This is seen at different ages during development, early in life. But we can see this from the first day of life. Even in preterm babies, they behave differently towards sugar if you put them in their mouths as babies. So the hedonic expression to sugar is different already at birth. You see this during infancy. In adolescents, we see in neuroimaging studies that they respond differently to palatable food images. In neuroimaging studies, they activate more areas of impulse control. And basically, as a summary of the phenotype, they're more impulsive towards this type of food. They cannot hold back to having and eating this type of food. And we believe that this phenotype, this behavioral phenotype, contributes for the development of their metabolic risk, right? Contributes for them to having higher type 2 diabetes, higher cardiovascular disease, and other consequences later in life. These findings we replicated in many different samples, and they were independently replicated by other researchers and labs around the world in large cohorts. So it's really a phenomenon that it seems to be very clearly happening. We're also able to establish an animal model that shows exactly the same features. And we're talking about translational studies, so this was very important in our lab. We have this animal model of caloric restriction during pregnancy. It's a strong caloric restriction, it's very powerful. The pups are born small, they catch up on growth before weaning, and they also have preference for high palatable food, rich in sugar and fat in adulthood. Next. So that's the framework that we'll work with. So early life adversity alters executive functions and impulsivity, cognitive flexibility. This influences palatable food intake, but it has other consequences too in behavior. And based on animal findings that I'm going to detail a little bit later, we believe that variations in dopamine neurotransmission in the brain, dopamine is this neurotransmitter involved in responsivity to the environment. And when you have values in the environment, for example, I need to work that much to receive that reward, dopamine is involved in these calculations. How much I put of effort here to receive these value as a reward or as a gift. And also involved in learning. So, of course, based on the phenotype that we saw, we thought, okay, dopamine is a very good candidate to explain these alterations. But considering that we're talking about low birth weight babies, we know that they're born with altered glucose metabolism and variations in insulin function. You remember that these babies are born with a smaller pancreas. They have less number of beta cells in their pancreas, so they produce less insulin. And this leads to a whole alteration in insulin sensitivity that they carry throughout their lives, right? Alterations in insulin sensitivity is a feature of a baby born small. And we thought that these alterations in the brain would influence dopamine neurotransmission and end up affecting the behavior. And I say that because the dopamine neuron in the brain has insulin receptors. And insulin acting on these neurons in the brain is known to influence the behaviors that we're talking about here, food preferences, impulsivity. So we thought that this was a good working model for us to explore and understand this relationship between early adversity and altered behavior from a neurobiological perspective. Next. So that's the rodent model that I spoke to you about. As I said, it's a 50% caloric restriction. So the pups are, of course, born small. At birth, we cross-foster them. So what it means is that all the pups are adopted by normal moms when they're born. We do this to ensure that the undernutrition happens only prenatally, not postnatally. So everybody is fed and breastfed by control animals. And in adulthood, you see at the bottom there that they prefer to eat more palatable foods, especially at night where rats eat more. This is coupled with an increased impulsivity towards palatable foods. If they have the choice between an immediate small reward or a delayed large reward, they go for the small one quickly. They cannot wait compared to control animals. So, and this is something that we saw in children that were born small for gestational age too. Those classical marshmallow tasks that the kid has to be waiting with little hands on the table until someone rings a bell so that they can eat the sweet reward. Small for gestational age kids at 3 years of age, they just cannot wait. They go there and eat the marshmallow quickly. So here is a good example of a translatable finding between the animal model and the human situation. Next. We also saw that these animals have a particular way of releasing dopamine in dopamine terminals, so the prefrontal cortex and the nucleus accumbens. You see on the left that the FR animals release less dopamine. At the 0 mark there, we gave them palatable food, and you see that the controls have a progressive increase in dopamine release in response to palatable food in the cage, and the FR animals don't respond. We also see that the time to reach dopamine peak is higher for the FR model in the other plot. It's the same when you give them rat chow, so normal food, but when you give them Fruit Loops, there's a delay in releasing dopamine in the accumbens in these animals. And this is completely reversed by an injection of insulin before the task. So again, strongly suggesting that is the modulation of dopamine by insulin is influencing this behavior and these outcomes. Next. We recently did this experiment that is doing genome-wide RNA sequencing from the brain of these animals. Genome-wide means that we took all the genes from the genome and we measured their expression at that moment when we collected the tissue. And what we can do when you have these, I don't know, 30,000 genes measured in the brain is that you can see which genes are working together or are being co-expressed in the tissue. So there's this mathematical way to do this that we call weighted gene co-expression network analysis. And it's a multivariate analysis, and we ask the data to tell us, okay, do you see gene co-expression networks in the tissue? And we found this network that is associated with the low birth weight condition, the food restriction condition. And if you ask another software to tell you, okay, what does this network represent, which processes it is involved in, and what's the point of this network in the brain, it is enriched for, you see, role of stress in obesity and type 2 diabetes, inflammation, the development of dopamine signaling, and response to insulin. So it's everything that we are speaking about. We can see also at the gene expression level. And this, I forget, this is in striated, yes. Yes, next one, please. So a word about insulin, you know this well, but insulin is this hormone that is secreted by the pancreas. It has an important action in the periphery, putting glucose inside the cells. So it's important for the use of glucose in the periphery, but it also enters the brain. There's an active transport of insulin into the brain. And there are receptors for insulin everywhere in the brain, many different regions, as you can see in the picture there. In the brain, the neurons do not need insulin to make use of glucose. As you know, there are glucose transporters that do that in the brain. But insulin is very important for neuromodulation in the brain. So it modulates neurotransmission, like dopaminergic neurotransmission, as I was telling you about. In regions, for example, like in the hippocampus, there's an abundance of insulin receptors there. And you probably heard about the Alzheimer's disease being called type 3 diabetes, because we believe that there's a role for insulin in influencing dementia. And so insulin in the hippocampus has a role in memory and cognition like that. In other areas, like the ventral tegmental area, it will influence dopamine neurotransmission in striatum and the PFC as well. So it is involved in all these behaviors like impulsivity, memory, learning, cognition in the brain. Next. We see reflections of this in our work. So this is the work done in adolescents in Brazil. And we saw that the HOMA index, which is a measure of insulin resistance in the blood, you measure glucose, insulin, and you do a calculation, is associated with the memory of a previous snack that they had, and also with brain activation in areas associated with attention and focused attention and things like that, like the prefrontal areas there and some visual areas too. Let me see if that's what I need to say here. Next, please. Okay, of course, we wanted to test this in larger populations. And, you know, in children, it can be challenging to collect blood and to collect blood multiple times for studies. Depending on the ethics committee and the size of your population, you can do that. Some other times, this is more challenging. In this case of this study, we had access to the MAVEN cohort, which is a Canadian-based cohort here, where we had data collected from birth until 6 years of age, later too, but consistently until 6. And we couldn't collect blood from these kids, like it was never approved. So, but we had genotype, which is funny, right? You cannot collect blood, but then you have the DNA. But anyway, we wanted to use this data to understand better these relationships between insulin and behavior, especially in the context of early life adversity. So here we used a classical technique that is the polygenic risk score, is that when you sum the small effects of many different genetic variants into a score. And then your score reflects variations in the risk for a certain disease or trait. You know the genome-wide association studies where you select gene variants that are associated with risk for a condition. For example, you run a GWAS for depression, then you identify, okay, all these are the top 1,000 variants that are associated with risk for depression. You run a GWAS for type 2 diabetes, these are the variants associated with diabetes. GWASs are amazing and revolutionized the way we think about genetics in the last few years, but they are also very particular to the population that you have. And most of the GWASs were run in adults. And here we wanted to test the effect of these scores in kids. So we needed to adapt a GWAS that was done in adults to children. And the way we did this was to slice an adult GWAS into 100 slices, and then select the slices that were mostly significant for children, testing in the AUSPAC cohort that had both genotype and fasting insulin measures. So we're able to test, okay, is our score really predicting insulin? Like, does it work if I go from genotype to insulin directly? So we use AUSPAC to train this. And then we created a score that was filtered for children in our Maven kits. It's basically a filter. That's why you see the strainer there. It's really to select the gene variants that were highly associated with fasting insulin in children specifically. Next. Then when we combine these variants into a score, we looked at an interaction model between the genetics and the environment, the early environment. Only impulsivity, again, using the marshmallow test that we call the snack delay task in Maven. Next. And we see an expected interaction. You see in the x-axis, as you go towards the right, there's an increase in adversity. Here we are looking not only at birth weight, but also other variables in the environment that are associated with poorer outcomes. For example, low socioeconomic status, exposure to violence, altered maternal metabolism, poor family function. So we sum them up in a score of adversity. And in the y-axis, you see the performance in the task. The lower the score, the more impulsive the child is, then meaning that they cannot wait for the sweet reward. And you see that as adversity increases, there's an increase in impulsivity, so a lower score, only for children with a high polygenic score for fasting insulin. So only for children who have a genetic background associated with risk for hyperinsulinemia. So again, showing a relationship between early life stress, insulin or genetic risk for altered insulin function, and impulsivity early in life. Next. We also run this study that is called Mendelian randomization. Probably many of you are familiar with this type of study. It's done using GWAS studies, and what it tells us is that you find it suggests causal associations between different features. For example, through GWAS studies, you can run a Mendelian randomization and find classically people have done this, like associating low levels of vitamin D with the diseases associated in bone and, you know, smoking and cancer. These associations that we know at the population level. Mendelian randomization is a good method to prove this in a empirical way. So we decided to use this technique to search for associations between insulin or fasting insulin levels and impulsivity using GWAS data. And we used sex-specific GWAS data, meaning that we run GWASs separately from males and females. And after all these calculations, what the study told us is that specifically for women, there was an association between fasting, insulin, and impulsivity only for those exposed to adversity, not for those that were not exposed. For males, the analysis was not conclusive, so we cannot say if there was an association or not with the data that we had. But for females exposed to adversity, this was very clear, suggesting that the association is there, and also that sex differences may be in place as well. Next. Yeah, you can go next, please. So yeah, we also wanted to, okay, it's nice that you can see, you know, associations between fasting insulin or genetic risk for fasting insulin, but you're always talking about peripheral levels of insulin there. That, yeah, insulin resistance in the periphery is interesting, but insulin resistance is also very tissue specific. And in the brain, insulin resistance will be brain region specific. You can have insulin resistance in the hypothalamus, but not in the hippocampus. And yes, in striatum, but not in other regions. And then the behavioral phenotype that you'll have as a result of that will be dependent on the region. So we want, okay, how can we test variations in brain insulin sensitivity or function only with this data that we have? We can barely collect blood, right? How can we do that? So we spent some time trying to create polygenic scores using the genotype data that we had to help us understand this. Next. So most of the genome-wide association studies, you heard about this in the news, right? Oh yeah, there was a new gene found for depression, new gene found for type 2 diabetes. We always find a new gene, and you can think, if you think about the synapses in the brain, I circulated there one that could be, okay, the gene associated with the biological process that we want to model. Next. But the thing is that that gene is not working alone, right? You see in the synapses so many other molecules and proteins working for the synapse to function as a process, right? So for this work, we are based in this gene co-expression network principle that suggests that genes are co-regulated by other genes. Therefore, genes that are expressed together are working together in the same biological process. So by measuring or by establishing co-expression networks, you are also identifying genes that are working together in the same process. Next. So that's basically what I mean. Like, you can represent all these molecules together in a network based on co-expression of all these components of the neurotransmission here represented here. In the same network. Next. So what we did was to use information from this network to calculate a polygenic score that is similar to the one that I used in the example before, but instead of bringing the genetic variants from a GWAS, I'm bringing it from the network. So supposedly variations in the score will represent variations in the expression of this network. And because gene expression is tissue specific, the score is also tissue specific, right? Next. So this technique allows us to have a variable in my data set that represents variations in the expression of this network. And then I can calculate this in large populations of people that have genotype, right? So I'll explain to you how we do this, but it's basically that's the basic idea. I put the QR code there in case you want to see studies that we published on this. I'll give you an example today. Next. So the basic difference between our polygenic score and the classical one is that the classical PRS that I told you at the beginning that we used as well, it selects variants or SNPs based on a GWAS. it reflects the genetic risk for that condition. In our case, we are using genetic risk for high fasting insulin. It could be genetic risk for depression, it could be genetic risk for type 2 diabetes. It depends on the GWAS that you used as a base. And this is very important, and it tells you genetic risk, is heritability, right? That's G in our G by E story. Our expression-based polygenic score, the SNPs are selected based on a co-expression network, on a network that we identify first using genome-wide expression data. And it will reflect individual variation in the function of the network in the specific tissue. So it's tissue-specific, it's based on gene expression, and is related to the function of the network. Next. So, okay, bear with me there. But it sounds complicated, but it's not really. What we do is first to identify a gene co-expression network. So I take like a piece of the brain, of course, in animal models. We measure the expression of all the genes in the genome. And then I ask this WGCNA to tell me, okay, do you see networks associated with being born small, for example? And they give me a network, a list of genes. I convert this to humans. I map all the genetic variants in these genes. And then I just calculate a score from there. How do we calculate the score from a list of variants? We're just doing matrices, like multiplying matrices one by the other. We have the genotype of my population here. We have the weights that I'm going to attribute to each one of the genotypes, and I multiply everything together. This part of the calculation that seems complicated is actually very classical. It's the classical polygenic score. The only difference of the technique is the way we identify variants. It's based on a co-expression network that represents a biological process. Next. You can go next. So what we did here was to select these two brain regions where the terminal of dopaminergic neurons are and where insulin modulates neurotransmission too, so striatum and the prefrontal cortex. We did this in animal models. We extracted the list of genes, converted to humans, and did all the calculations and calculated this expression-based polygenic score of the insulin receptor in this brain region. So we're looking at genes that are co-expressed with the insulin receptor in the brain, not in the whole brain, in these two brain regions there. Next. So that's the network. This is older work now, but it's important that I explain this so I can show other stuff for you. This is the network. You see it's pretty connected, meaning that these genes are co-expressed in these brain regions indeed. Now, if I take the same number of genes from a regular GWAS, the classical approach, and I try to do the same drawing here, they don't really form a network. You can go next so we can show that. You see they're not connected, like it's just a few of them are linked, but the classical approach doesn't really form a network on its own. And if you take a random list of genes, they also don't form a network. So it's really only the co-expression network as a base is that allows us to calculate the score like that. Next. Again, this is older work, but when we do this in these brain regions, prefrontal cortex and striatum, it predicts addiction and impulsivity in humans. And if we calculated this based in a different brain region, the hippocampus, as I told you, is going to predict cognition and risk for Alzheimer's. So it's aligned, the place where you choose to calculate your score, it's aligned to the function of insulin on that brain region. So this is proof of concept work that was very important for us to understand that the score really reflected what we wanted it to reflect, like we imagined it was reflecting. But then we could go to the next slide. So we're going to see what we did with this. And that's where I want to show you this is newer work that we're doing. Of course, we wanted to see associations with metabolic risk and long-term effects of early adversity. In the x-axis here, you see a score that combines negative influences like the orange stuff to positive ones, so kids can vary between, you know, being raised in a poorer environment, more adverse, or more protected one in the blue side. And then the y-axis is probability of having obesity in adolescence in the ALSPAC cohort. You see, as adversity increases towards the orange part, there's an increased risk for obesity only in those that have a high polygenic score for insulin receptor in these brain regions, and markedly with sex differences as well. Despite the fact that both sexes go in the same direction, this is significant for the girls in this age. Next. The same interaction we see later on in this large population, more than 500,000 participants of the UK Biobank. In this analysis, we have more than 60,000. Same thing. As adversity increases towards the orange there, we see an increase in visceral adipose tissue volume, especially for those that have this high expression-based polygenic score of the insulin receptor in the brain, and especially in girls and women, of course, in this. Next. Yeah, I just brought this super new work here. Is just telling you that, again, another proof that this is tissue specific. Here we calculated the same thing, but just changed the initial tissue. So here we're using the VTA, which is this little part in the back of the brain, where the dopamine neurons are, the body of these neurons is there. When we do that, again, we see in the x-axis here is birth weight. As birth weight decreases, there's an increase in impulsivity, especially for those with a high PRS from the VTA than for the insulin receptor. So again, showing that the score changes as you change the region, but consistently predicts what we expect. It predicts, like, interacts with early life adversity and predicts impulsivity and risk for metabolic disease. Next. How are we in time, Chris? I can skip this.
Pode continuar tranquila, Paty.
Tá, obrigada.
A professora Guadalupe não vai poder estar conosco hoje de tarde, então a gente tem tempo liberado.
Opa, então vou contar mais coisas.
Que ótimo.
Great, so we can talk more about newer stuff. So this is, again, newer work. Now we're kind of combining the initial work that I was telling you about with the polygenic scores, and I'm trying to capture here the molecular signature associated with early adversity and brain response to insulin. Next. So, okay, now I'm going to go super slow because we have time. The model is the same as I showed you before. We start at pregnancy day 10, and then we expose the FR, the food-restricted animals, to 50% caloric restriction. What we do is to measure every day of the control animals, what they eat normally. Then we calculate half, and then that's what we do give to the food-restricted group. So then we do that. At birth, all the pups are adopted by control dams. So you know that the intervention happens only during pregnancy. We wean then at 21 days. And in adulthood, we are ready to do the experiments. In this case, what we did was to inject them with insulin and then collected the tissue. We wanted to see the brain response to insulin in terms of gene expression. So we did this, injected insulin, waited 30 minutes, collected the tissue, and then did the genome-wide RNA sequencing. So I measure gene expression, the entire genome, in this case, in the prefrontal cortex of these animals at P90, so in adulthood. After doing that, you can use this data to identify these co-expression networks using the WGCNA that I told you about. And that's what we did. Next. So when we do this, it's small in the little square there, but you see like there are many networks. Some are more significant, more red or more blue, and some are less. But there's one that is particularly significant that I put there in the green square. This module, this network, is associated both with being born small, or the FR condition, as well as response to insulin in adulthood. So it's a module that is interesting for us because it seems to be capturing this signature, okay, of who was born small, and also an altered or a differential response to insulin. This network represents that. And that's what you see here in the network. It's a good size network. I don't remember how many genes here, but it's a good size. It's not that we're talking about only a couple of genes. It's a good size of a network. They are connected. You see, like you see the co-expression in the gray lines. And if I ask the software to tell me, okay, what does this network mean? What is it doing, right? What are the processes associated? It is associated with oxidative stress, statistically significantly linked to oxidative stress, as well as diseases like panic disorder. So suggesting that this network is involved both with metabolic outcomes, like oxidative stress, as well as mental diseases like panic and anxiety, right? So the network seems to be relevant for both physical and mental health related problems. Next. Now, of course, I wanted to take this network and calculate the score, right? So that's what we did. We used the information from this network that came from the animal model, represented this response to both being born small and responding to insulin in a specific way, transformed this to human genes, identified all the gene variants in these genes. Then we did the math, the hard math there to transform this into a score. So it became a variable in my data set. Variations in the score represented variations in the expression of this network in the prefrontal cortex. So individual variation, how people vary in this naturally in this network that represents processes linked to being born small and responding to insulin. Next. I wanted to test this in relation to both mental diseases and cardiometabolic diseases, because that seems to be the functions that the network represent, right? And for doing that in adolescents here in AUSPAC, using data from this cohort from the UK, we created this analysis where we identified children that had higher values of risk for metabolic disease and for mental disease. So what I did was this cluster analysis and asked the software again to tell me, okay, which kids have higher risk for metabolic disease as well as higher risk for psychiatric disease at this age. And we use questionnaires like total difficulties in the SDQ, which is a mental health questionnaire, depression score, anxiety score, the HOMA index, which is that measure of insulin resistance in the blood, as well as weight circumference. And when I do that, there's a group of children, the blue ones, who have higher levels in every single one of these measures. And there's a group of kids, the green ones, who have lower measures. So the blue ones have higher risk for comorbidity, and the green ones have lower risk, right, in many factors together. Next. I told you about the differences again, but just to keep in mind, the PRS, the regular polygenic score, represents genetic risk. The EPRS represents variation in the expression of that network. Next. So the interesting finding that we had here was an interaction between the genetic risk and the expression-based polygenic score I'll guide you through. So in the x-axis, we have variations in a polygenic score for major depression. So this is classical genetic risk for depression. As you go towards the right, you have more genetic risk for being depressed, higher PRS for depression. And then the y-axis is the probability of comorbidity, being the blue or the green group that I explained you before. And you see, as the risk for depression, the genetic risk for depression increases, there's an increased risk for this comorbidity during adolescence. But this is true only for the people who carry a high expression of this network in the PFC. So you see, the expression-based polygenic score is making the genetic risk more accurate. We're selecting people that have really a higher risk that goes beyond the genetic risk and explains more of the variance of the risk for comorbidity here. Remember that this network, the EPRS group there, we're representing both being born small and responding to insulin in a particular way in the PFC. So this tells me that our score is actually capturing this signature of being born small, responding to insulin in a particular way in the PFC, and that this is relevant for the risk for comorbidity between mental health problems and metabolic disease. Next. I think I'm getting to the end. Yeah, it's just a summary figure of a review that we published a couple of years ago, where we look for this interaction between genes and environment on this trajectory of growth and neurodevelopment and diseases and aging. You see, what's relevant for the thing that we are discussing today, translational studies, is that we come from the animal model. We can model adversity in a very precise way. In the model there, you can control exactly the day of the pregnancy where you start the intervention. You can collect tissue in a very precise manner from a very precise region and measure gene expression that, and then use this to inform the calculation of scores in humans that can be applicable to large data sets, and you can see these effects at the populational level. I gave you one example here of the thing that is interesting to me, but of course this technique can be applicable to other areas and other pathways and other research questions as well. Next. Yeah, I think it's just another summary slide of what we spoke about here. This is another review that we published recently, talking about sensitivity to early adversity and how insulin probably makes you... Insulin is an important moderator of this association between early adversity and long-term disease. I think the next one is the last one. Yeah, so just finalizing, to thank for the invitation to be here. It's so nice to see you again, long-term collaborators and colleagues here, as well as the new people that are part of the course. Always thank the funders as well, and of course the students, because without them nothing happens. And this is an older picture of the lab, but still most of them are here. And yeah, thank you. Happy to answer the questions.
Bueno, muchísimas gracias, Patricia. Yo como neonatóloga me parece alentador encontrar un poco de tu investigación, un poco la expresión que nosotros lo vemos clínicamente en el seguimiento de estos niños que han sido prematuros o bajo peso, que tienen trastornos de conducta y que a veces no lo podíamos explicar por una causa química. Y ahora que ustedes pueden, desde la investigación básica, linkearlo con el dosaje de insulina, tal vez, bueno, mientras se generan preguntas de la sala por ahí, me gustaría saber si ustedes pudieron asociar alguna morbilidad específica que se asocie más con la insulina, como para uno buscarlo clínicamente en estos niños? ¿Pudieron encontrar algún valor más alto de insulina en alguna morbilidad específica dentro de lo que es trastorno mental o obesidad o algo de estos niños que son de peso?
Sí, no tengo un valor específico, no, pero la asociación con las doenças específicas como depresión parece ser muy, muy importante. I'm going to jump again back and forth to English because sorry about that. Okay. But the challenge with insulin values is that it fluctuates during development too, and this is normal, right? For example, right before catch-up growth, it's an insulin-dependent process, so you have to have higher to be able to grow. Same thing before the growth spurt during puberty. So this physiological changes that insulin has during development, it becomes a bit difficult to work with the peripheral values and establish cutoffs and stuff like that. But yeah, hopefully we'll be able to map this trajectory and have this established for clinicians. Yeah, that would be great.
Yeah. Paola?
Gracias, Patricia, por una presentación siempre tan inspiring. Every time I listen to your talk, I get different approximations, probably because each one of us has been working all this time on interrogating hypotheses and trying to explain how things occur. With new information, with new ways of thinking things. So I was thinking, I have several questions. One, very naive question, right? Is insulin resistance in the brain the same mechanisms for insulin resistance in the periphery? So should we just move everything in the brain equivalently? Is the receptor, is the signaling downstream of the receptor affected? How is the brain insulin resistance occurring?
That's a great question. And the answer, of course, is more complicated. It's not. It depends. Even in different brain regions, you're going to have one or other pathway being more relevant for the action in that brain region. So, for example, the AKT pathway versus the other pathways, it will depend on the brain region. So, of course, it makes everything way more complicated, but also way more interesting to study and to investigate. Yeah.
Sure. Can I follow up with the questions? Because I have like 3 or 4 questions. You just stop me, okay? If any of the other students ask questions, then it's okay. I also had another question. I find it so interesting how you can extract information from the interaction of the polymorphisms in genes and the expression of the genes in the animal model, and you export this information. You ask questions to the model, and you have responses that are extrapolable to the human, right? And I was wondering, those data that you get from the children's of the 90s, the ASPAC and the UK Biobanks, that you have samples from adolescents or adults. In those samples, do you have the option to look at the lineal, to see, let's say, the population when it's 15, you have this score related to adversity and metabolic insulin resistance, right? When you look at them at 20 or at 30, do you keep, is this, because this is really, if you think at the human beings behind these numbers, you can see, is this permanent? So somebody that has suffered adversity early in life has more risk of having metabolic disease. And is this going to be worse and worse, or are you able to do some kind of interventions that you can modulate?
That's an excellent question, Paola. And you know, from the data perspective, especially AUSPAC is a beautiful data set because they track them over time from birth to adulthood. They're now, I don't know, 30 years old, and they have multiple data points of measuring, for example, BMI, mental health, and other phenotypes. It's an amazing cohort to work with. The UKB is not longitudinal, it's cross-sectional, but it is so rich in terms of phenotype and genotype and neuroimaging and size, because it's 500,000 people, that, you know, you can find ways around it, and they have over a span number of years, they were able to follow these individuals. So it's pretty useful as well. Now, the question that you ask has also other levels that we're trying to tackle as well. So today I showed you information from a network that we collected at P90 in the animals, right? So this is a network that is responding to both birth weight and insulin in adulthood. Now, we also collected tissue earlier from P0, P21, and there are other networks that seem to be more relevant in other time points during development. Now, there's a very talented postdoc in my lab that is mapping this, like, what does the network that you identify at birth or during adolescence or in adulthood, do these scores predict trajectories, or how is the relationship between these different time points of gene expression that we can only bring from the animal model, because I don't have these measures in the brain of the people, right? I can only translate from the animal model, but still we're tracing exactly that, how the changes in the network over time map or can predict diseases, or are they associated with resilience, maybe, right, to these outcomes? That's what you're asking, like, can we envision some sort of intervention there based on these associations? So yeah, next year when you, I can come back and tell you about it.
¿Alguna pregunta del público, de los estudiantes?
You can ask them in Portuguese, Chris, because maybe they're shy.
Sim, posso falar em português e posso intentar em espanhol um pouquinho.
Não, está super bem o espanhol de Pati também. Pessoal, fiquem bem à vontade, tá? Para perguntar no idioma que vocês se sentirem mais à vontade. Eu vou aproveitar, então, esse momento para fazer uma pergunta para a Pati. Primeiro, Pati, muito obrigada. Tuas apresentações sempre geniais, e tu sempre nos traz os dados, assim, contando toda a tua história, a gente vai acompanhando, e é realmente muito lindo, assim, ver todo esse crescimento e essa complexidade, né?
Obrigada.
Que tu vai agregando a cada trabalho que tu faz. E eu estava pensando justamente no aspecto translacional. Vocês chegaram a fazer algum tipo de comparação entre as estruturas cerebrais e o sangue para tentar fazer uma comparação com os resultados em humanos no futuro?
Sim, boa pergunta também. Nos ratos lá, a gente mesmo não fez, mas esse modelo é classicamente associado também com... Não, na verdade não é verdade. A gente chegou a ver também que eles têm hiperinsulinemia, né, os animais, e algumas coisas como ansiedade também a gente vê no comportamento dos animais. Mas, assim, para confirmar a questão da translação, né, porque tem um passo que eu não tive tempo de contar para vocês, quando tu tem a lista de genes dos animais para transformar para os humanos. So maybe I'll speak in English because just to make sure that everybody's on the same page. When you have to translate the gene list from the animals to humans, there are things that you can do midway to confirm that this gene list is also co-expressed in humans, for example. So there are validations that we do there midway that I didn't talk about today, but you can use, for example, post-mortem tissue data sets in humans to confirm, okay, these genes that are co-expressed in the animal in this brain region also form a network in humans. And so, yeah, you have to be careful in translation, because it can be poor if you don't do these things. But it's very important for the work to be able to go back and forth as you're suggesting and confirm these associations. Yeah.
Muito legal. Não temos nenhuma questão ainda aqui. Temos. Paola. Vamos ficar nesse bate e volta. Paola.
Okay. There's different ways to inspire the students. If they don't want to speak, there we have Margaret. Okay, I'm going to make it short, Margaret, so you can ask later. To inspire the students, to lose the being shy, you know. The other, you know, Patrícia, we've been in contact because we have this small study. We got a small amount of funding from the triada between Tecnológico de Monterrey, Colombia, and Chile, where we're doing early adversity study. We're beginning at the end of September, finally, because we have all the ethics approval. You know what that means? We've been since February with the ethics approval, and finally we have everything ready. We're going to take 80 dyads, mom, a caregiver, and the infant between 3 and 5 years old, boys and girls, from the public, how do you say, kindergartens.
Mm-hmm.
And we're going to take, they left to take a little blood sample from a few dots of blood with these small automatic analyzers that's going to give us some of the lipids, insulin, and glucose.
Mm-hmm.
So, wow, we're very happy with that. We're going to take hair to look at the hormones, as we have spoken before. And the psychology team is going to do a lot of instruments to measure adversity.
Mm-hmm.
But also, as one of the PI from Chile, which is a psychologist, Pia, is interested in the benevolent. I don't know in English if you say benevolent, but in Spanish you say benevolent exposures that could be protective. So they have this very new instrument they're trying to apply, because it's not only about adversity, it's also about what stands for resilience, because that is an opportunity to do interventions, right?
Yeah.
So so my question was, what do you think about sample size? Because we were going to have 80 adults, 80 children, so 160 subjects, dyads. We're going to have 80 parents and children in Colombia and 80 in Mexico, to have the idea was to have a scope of what's happening in Latin America in these topics. Yeah. So at the end, we're going to have 240 dyads. What do you think about these numbers? Are we going to be able, we're going to take DNA from the mouth.
Good.
So at one point we can talk about collaborating there. If we can look at some genetic marks, that would be fantastic. What do you think about the number 240? For us in the practice is a lot of work.
Yeah.
We're going to do a lot of measurements. We're going to weigh, we're going to take circumferences of the arms, of the, take the body fat. Débora Sabatelli is very happy. She's looking at this. I want to see those numbers. Well, what do you think about this? Are we going to be able to look at some of these scores and things, or is it too small? We need thousands of kids. How is it for the people that do clinical work? I see Fabíola, Suano, I see, other clinical people. Can we do this here?
You can, definitely you can. And the reason for that, many reasons for that. One, what you miss in sample size, you gain in depth of information, right? So clinical studies, even though sometimes we don't have the super large sample, they're so detailed, as you're saying, you're describing super detailed evaluation, that you have enough information to look at different markers and make associations. Now, polygenic scores also do not require thousands and thousands of people. Genome-wide association studies, yes. But when you're bringing this to calculate scores in your sample, then you don't need it, because the big discovery was done in the GWAS that is published in Nature. There, you use that information, you calculate the score in your sample, right? So this is fine. And the third comment there is that you can do things like I showed you here, that I had the GWAS, I validated this using ALSPAC data, and then I used in a small sample from MAVEN. My MAVEN sample was like 130 kids, but I validated the score using these data sets that already exist. So I don't need to do validation work in my small sample. I use something that I validated through other cohorts. And the world that we live in today is amazing for data sets, because there are many data sets that are available for free, right? It's not the case of ALSPAC. ALSPAC is kind of expensive to request the data. It's super worth it, I'll tell you. I don't gain anything to make propaganda, but, you know, having collected data myself, it's super worth it. Like, you pay and you have it there, and it's, you know, can do many studies there. But there are free data sets like ABCD from the United States, like 15,000 people, that you submit your request, you get the data, and you can work with it, and it helps you validate your score before you calculate in your sample there. So there are many ways to deal, and there's no like, I see tremendous value in small, very deeply phenotype studies, because we can take so much information from it. And today, coupling with these other tools, you can do like amazing studies there.
Excellent. Thank you.
Margaret?
Oi, boa tarde.
Oi, tudo bom, Margareth?
Tudo bem. Primeiro, eu não sei, a sensação que me deu essa aula, professora Patrícia, foi assim, de um pouco de emoção misturada com um pouquinho de tristeza. Porque quando você pensa em adversidades, e eu sei que você estudou ratinhos, mas a gente que lida com crianças que nascem em hospitais públicos no Brasil, em diversas situações difíceis, né, diferentes de norte a sul, eu fiquei pensando, às vezes a gente está em hospital em neonatologia e você pensa em questões clínicas, né? Abrigar tanto, nasceu pequeno para a idade gestacional, passou por ventilação, aquilo tudo. Mas não considera esses contextos outros, né? Que você colocou as diversidades. E eu fiquei pensando se há esse índice de adversidade neonatal, o que a gente poderia fazer, se há alguma coisa que se possa fazer para ir contra isso. Depois que já nasceu, eu sei que pode haver, então, fatores que vão estar associados a doenças no futuro dessas crianças, mas enquanto internados ou enquanto em follow-up, a gente já conhece algumas intervenções que a gente faz, né? Aleitamento materno, pele a pele, melhorar a ambiência. Mas há estudos que mostram, assim, com translacional com ratos, que a gente pode ir contra esses maus desfechos de uma forma realmente que a gente possa trabalhar e trazer para as questões de saúde pública, de um pré-natal melhor e de uma internação melhor para as crianças. Porque o que a gente tem são bebês realmente que nascem pequenos, malnutridos, de mães, às vezes, que vêm usuárias de droga, e deixam os bebês que passam 2 anos abrigados e que vão ser entregues à sociedade um dia. Sim. E é muito preocupante, porque no seu slide são 25% de uma população.
Exatamente.
E aí a gente se juntar as outras adversidades, eu não sei quanto por cento seria, por exemplo, eu moro em Fortaleza, no Ceará. Eu não sei quanto por cento da minha população estaria ainda num quadro pior, entende?
Sim. Sim, excelente questão e muito relevante e muito real, né? Palpável, assim. Os estudos que a gente fez tentando encontrar soluções ou artifícios que pudessem nos ajudar a reverter essa situação não trazem nenhuma novidade. Coisas que tu comentou. Breastfeeding, long-term breastfeeding, exercise. we explore other things like omega-3 consumption and things like that. However, there's also another aspect to adversity. Tu fica confortável se eu falar em inglês? There's another aspect to adversity that is individual differences in the response to stress. Now, it's not, we didn't talk about this today, but it's not 100% of the kids that are born small that will have problems later. There's individual variation there. Some kids will respond more or will suffer more than others. And if we give intervention across the board like that to everyone, first, we don't have enough manpower for this. And second, not everybody needs, because some of them will just do fine. We need to identify who's going to need really the intervention and who's going to respond and which intervention is the best for that child, for that family, for that reality, for that neighborhood, right? That's the challenge I find. Because, you know, if you go to a neighborhood and say, oh yeah, you have to read to our children, and the parents work, you know, 9 to 9 and they are struggling to bring food to the house and everything, like, oh, come on, like, it's just not feasible, right? So planning interventions that are real and feasible and palpable and empowering parents to be able to provide this to their kids, aligned to identifying who really needs, who's going to be suffering long-term consequences, and who will do fine, like, just leave them. That's the challenge. So we've been working on this in another project, trying to identify, and Paola knows about this work, is aligned to the project she was describing, trying to identify markers of stress responsivity in young children, to really come up with a battery of markers that tells, okay, this child was born small and she is suffering from stress, so we need to intervene right now. And also to have some sort of metric that you can track. For example, you propose an intervention, let's say parent-child group or something, support or nutritional intervention. You should be able to track, is this helping or not? Like, you cannot wait 30 years to see if it worked or not. You should be able to track. So we've been working on that, trying to develop this battery that helps us identify who needs, what's the intervention that is best for that child, and is it working? We have a long way to go still on this work, but I think that's what we need, you know.
Obrigada.
Obrigada. Sí. Fabíola.
Olá, boa tarde. Good afternoon.
Tudo bom?
Muito obrigada pela sua fala, Patrícia, muito importante, e inclusive nesta pergunta que a Margarete fez agora, né?
Uhum.
E eu ia só complementar uma coisa, que a Margarete é uma pessoa que está sempre nos nossos cursos, viu, Patrícia?
Que legal.
She always with us in our meetings, it's my pleasure because she's thinking about what she learning, not just with us, but with everybody, and is like improve the knowledge about what I can do, what I should do. Is exactly what we are thinking now is translation idea. I think I know some studies, cohort studies in Johannesburg, Linda Richer, and here in Brazil we have a very important study with Carol, Cris, what is the name? Recife?
Carol Leandro.
Leandro, and because it's important we have studies with our population here in Brazil, for example, because as everybody knows, we have a lot of problems together, but we have a lot of solutions together. So they show this type of studies, for example, Joanesburgo, África do Sul, Linda Richard and Carol Leandro in Recife. The results of these studies showed that it's possible to improve, yes, it's possible to improve the quality of life, and especially when we are involved in neurodevelopment and quality of life, especially with one thing, because it's interesting, not just one, of course, but what is important, I don't know in English is correct, but is a parentalidade, parenting. Somebody take care of the child.
Yes.
Somebody. If it's possible, the mother, the father, the family. If it's not possible, the school is important, the public policy is important. So the plasticity of the brain development, it's very important, and improving the neurodevelopment is it's a target. Yes. Especially when I think about the intrauterine problems. So Margarete, don't worry. Of course, we have a lot of problems, but what the studies are showing, especially with populations like us, double burden of malnutrition like this, it's possible to improve. But we need studies like that you show it for us today. Thank you so much.
Thank you, Fabíola.
Se eu puder falar ainda.
Por favor.
Com licença. Eu tenho estudado no doutorado a questão do aleitamento materno e o neurocomportamento de bebês. E quando a gente estuda em hospitais universitários, né, com populações de mais risco, você acaba vendo uma realidade mais, né, na sua frente. E eu vejo as mães aqui em Fortaleza, por exemplo, nós temos ordenha à beira do leito. Então, a mãe entra com um copinho, ela ordenha o leite, oferece ao bebê assim que ela ordenha. E outros tipos de ordenha e o leite é ofertado. E eu me surpreendi com a minha coleta, né? Porque eu coletei uns 2 anos e tem mães que ordenham litros e litros de leite. Numa situação de estresse altíssimo que elas vivem. E isso me deu uma outra visão dessas mães que estão lá. Eu me emociono um pouco, porque é difícil ordenhar leite, né? Eu sou mãe e eu sei que é difícil. E isso mostra uma força de vontade, né, de melhorar os seus filhos, de ter um bom desfecho. E às vezes eu vejo uma falta de vontade dos gestores hospitalares. E acolher essas famílias, como a professora Fabíola falou, na parentalidade, né? Na ausência da mãe, na ausência do pai, isso dificulta um pouco esse desenvolvimento do bebê. Porque nós, profissionais, não substituímos. Podemos até tentar ter um cuidado mais afetivo, mas não substitui. E às vezes falta um espaço para essa família estar dentro da unidade. E o impacto disso no bebê, né? A questão do desenvolvimento, de uma alta mais precoce. Isso me toca muito. Acho que é por isso que a professora disse que eu participo, mas é porque realmente me toca as ações que são leves, né? Que não são caras.
Exato.
E que a gente pode estar fazendo. Mas é um desafio muito grande, porque requer vontade de pessoas. Acho que é mais isso. Obrigada.
Que bom que tu está falando isso, Margarete. Eu quero muito ver o teu estudo publicado lá, porque eu acho que isso é muito importante e contribui muito para o avanço da área. A resistência não é só a nível de gestores, eu acho que é uma resistência que existe em vários níveis. Existe também na minha área, na pesquisa, na clínica, também existe. O aleitamento materno é uma coisa assim que a gente que trabalha com isso sabe dos benefícios, mas parece que a gente tem que estar sempre provando, né? Então, é uma coisa que a gente precisa colocar na rua, assim. A gente publicou um trabalho agora, o último que eu publiquei aí, em que a gente buscou identificar os fatores que estavam associados com o neurodesenvolvimento em bebês primeiro mês de vida, com a habilidade de track, né, um estímulo, então, assim, fine motor skills, e a gente mediu uma série de coisas, doenças maternas, o próprio cuidado materno a gente mediu, e tipo de brinquedo que tem em casa, nível socioeconômico, mas os fatores que mais predizeram a cuidade do nenê, a habilidade, são fatores metabólicos e nutricionais do leite materno, né? Então, para ver assim, uma relação direta com o neurodesenvolvimento do bebê, que é bem o que tu está dizendo, né? E a gente que trabalha na clínica sabe que eles precisam desse apoio, esse que tu está falando, né? As mães conseguem, mas elas precisam do apoio, precisam aprender a fazer. Uma mãe de primeira viagem precisa aprender, né? Tem que estar alguém ali para dar uma força, né? E, infelizmente, tem bastante resistência, assim, para prover esse serviço, assim, né? Então, na minha área de pesquisa, assim, né, acho importante que isso seja publicado e esteja essa palavra na rua, né? Mas continuar o serviço de formiguinha na clínica lá também, cada mãe que se consegue é mais uma, né? E porque isso é muito importante.
Eu tenho mais uma questão para a Paty. A Paty tem uma tradição de acolher a muitos estudantes de toda a América Latina. Eu não sei o número de estudantes que a Paty tem agora, mas da última vez que eu estive no laboratório dela, era como essa disciplina, se falava espanhol, português, inglês e não sei mais quantos idiomas no laboratório. So, Paty, which suggestions you can do to all these students? Now we are in 7th one here, to be prepared to work in a high level as in your lab, in a highly productive lab around the world.
Great. No, this is a great question, Chris, and thank you for saying that. I'm proud of the lab being so multicultural there here. But I think that the top feature of a student to work in a lab is something that is very dear to us from Latin America, is to work very hard and to be passionate about the work. That's the top feature. I receive students in the lab that never heard about genetics before and never heard about the polygenic score, and come to the lab, stay for a while and learn, and they are able to work and do the work just because they're so passionate and they work so hard, right? So there's no limit for someone that works hard and is passionate. Things like language, of course, is important. It facilitates the work. It's faster if you know. But the main thing is to work hard and be passionate. That's the top.
Great, espetacular. Temos mais alguma questão? Temos mais alguma pergunta para a doutora Patrícia? Nesse momento, não. Eu gostaria de dar um aviso, a professora Guadalupe, infelizmente, não poderá estar com a gente hoje de tarde. Ela teve um inconveniente e não pode estar com a gente, mas ela já nos mandou uma mensagem lamentando e pedindo desculpas, então, a vocês por não poder estar aqui hoje, e já se comprometeu a agendar um outro horário para dar, então, essa aula, e a gente vai manter vocês informados assim que a gente tiver, então, a agenda correta com a data. Entonces, doutora Guadalupe no pudo estar hoy con nosotros, pero les manda muchos saludos y nos dijo que luego que pueda ya va a agendar un otro horario para la clase y mantenemos entonces todos informados para que tengamos esta clase con la doctora Guadalupe Estrada en el futuro, en los próximos semanas o próximos meses. Estamos pensando incluso en hacer un webinar, ya aprovechando, ¿no, Eri? Los webinars del DOHaD latinoamericano y incluirla en uno de nuestros webinars. Entonces, stay tuned. Tenemos ahora a la próxima semana un webinar, un simposio en México, el día 19, que estaba proyectando ahora al principio, probablemente ya lo recibieron por correo electrónico. El día 27 tenemos el lanzamiento del website del DOHaD, del Congreso DOHaD Internacional, entonces va a ser el Congreso Mundial que va a pasar en Buenos Aires, la beautiful, hermosa Buenos Aires de Débora, y todos allá entonces en 2025, justamente, precisamente en septiembre, de 7 a 10 de septiembre estaremos allá. Y Tenemos ahora en octubre un webinar hablando de doenças emergentes, enfermedades emergentes en Latinoamérica. Hablaremos de Zika, fiebre Oropouche, todos estes vírus que afectam especialmente a las embarazadas, y va a ser el webinar de octubre con la doctora Julia Clark, que es de la Universidad Federal de Rio de Janeiro. Entonces, tenemos muitas atividades hasta el fin del año, todos muito bem-vindos. Eu gostaria agora de deixar o espaço aberto para todos se manifestarem, e depois, no final, então, a gente faz um fechamento, e agradecer muito, muito a participação da Paty. Sempre, Paty, te ouvir é genial, porque a gente fica pensando em quão orgulhoso a gente é, sabe, de ter, assim, os teus trabalhos, de poder se inspirar em toda a tua história de vida também, que é realmente maravilhosa. Então, parabéns e muito obrigada por compartilhar conosco.
Obrigada, Cris. Obrigada pelo convite.
Erika. Parecía que había levantado la mano.
Parece que quiso reaccionar. Yo creo que quiso reaccionar y apretó la mano. Ok. ¿Queremos cerrar entonces esta sesión?
Pues tenemos una pregunta,
no sé
en verdad, ahora. Leo pregunta, ¿hasta cuándo va la disciplina? Hasta ahora, Leo. Estamos terminando justamente hoje a disciplina, pero... fala em português, né? Então, a gente está terminando hoje a disciplina, Léo, mas a gente vai ter ainda a aula da professora Guadalupe Estrada, e a gente vai enviar para vocês a data correta, porque a gente ainda não tem a confirmação da data em que ela vai ocorrer, tá bem? Desculpa, Paola, perdão, Paola, por favor, sigas.
No, está bien, perfecto. Esta es la última actividad, sí. Jennifer, es la última actividad. Se cierra el curso con esta sesión de hoy día, y la profesora Guadalupe Estrada va a presentar en el formato de webinar, no como parte del curso, ¿cierto? Los estudiantes me imagino que están preocupados porque tienen que contestar las preguntas, etc.
Exacto.
Sí, hasta aquí queda. Me imagino que la grabación ya se acaba, y entonces queremos agradecerles a todos los participantes del curso por haber participado. Tuvimos prácticamente 100 estudiantes, un poquito menos de 100 estudiantes participando en las distintas sesiones, así que agradecer a todos por el interés, que el capítulo latinoamericano de DOHaD siga creciendo, que todos los interesados puedan encontrar un espacio donde hacer sus investigaciones y contestar sus preguntas, sus hipótesis, donde se puedan discutir en un espacio de confianza, donde puedan, en el fondo, interactuar. Los esperamos a todos el próximo año, en un año más preciso, en Buenos Aires, nuestro Congreso Mundial. Así que nos estamos viendo en los distintos webinars y nos hablamos hasta el próximo año, ¿sí? Para nuestro Congreso.
Los estaré esperando acá en Buenos Aires, todos, junto con Alejandro.
Eso sí, espéranos, muy bien, con unas buenas parrilladas y con unas... Eso.
Estamos preparando todos, comprometidos con tener un excelente congreso. Creo que van a quedar muy satisfechos. El programa científico es sumamente interesante, sumamente atrayente. Contemplamos la problemática desde todas las perspectivas que hemos podido. Paola, Alicia Mota, Gloria han trabajado muchísimo para que así sea. Y bueno, y estamos preparando para que Buenos Aires esté radiante, florecida como suele estar en septiembre, y con muchas ganas de recibirlos a ustedes. Esperamos estar como anfitriones, estar a la altura de las circunstancias.
Excelente. Gracias a todos, Cris, Fabíola, Erika, como coordinadoras del curso, Débora, por estar hoy día coordinando, y por supuesto Patrícia, nosotros decimos en Chile la guinda de la torta, ¿sí? The cherry on the top of the pie. Thank you very much, Patrícia.
Thank you, Paola.
Eri, tenías la mano levantada cuando caíste, ¿quieres hablar? Después Fabíola. Y ahí Fabíola hace nuestro super cierre.
Perdón, una disculpa porque hemos tenido un chorro de problemas aquí donde estoy en la región, se está cae y cae la señal, también por eso Lupita, precisamente déjenme comentarles, un poquito transmitirles su angustia, ella tuvo que salir del instituto para ir a su casa pensando que iba a estar más estable la señal y sucedió el accidente, entonces estamos teniendo ciertos problemas, una disculpa, pero a todos los estudiantes, a todos los speakers, a Cristiane, Fabíola, Paola, ¿no? a todos los que han estado coordinando las charlas. Es un esfuerzo muy grande y DOHaD está creciendo. Estamos muy contentos porque el trabajo de cada uno de ustedes suma para hacer una ampliación de este concepto, de este conocimiento en todos los niveles, ¿no? Entonces, estamos muy contentas, muy agradecidas. Sigan esforzándose, continuando. Siempre decimos, vamos a alcanzar las metas, no importa en qué momento, pero las vamos a ir alcanzando. Entonces, no dejen de seguir luchando, participando, soñando y preparándose. Muchas gracias por el curso a todos, a todas. Y pues disculpen la situación al último, pero me da mucho gusto verlos aquí. Muchas gracias, doctora Patrícia, por su charla. Muchas gracias.
Bom, vou falar em português. Será mais fácil agora. Mas falarei devagar. Primeiro, muito obrigada pela presença virtual de todos. É muito bom saber que mais de 80 pessoas, de 80 a 90, 70 a 90 pessoas estiveram conosco, tirando os professores, que então a gente passa de 100 pessoas durante essas tardes, aprendendo juntos, ouvindo coisas novas, inspiradoras. We are inspired, of course. E mais do que isso, né, desse grupo, nós temos aí 80 pessoas, pesquisadores, alunos de pós-graduação, futuros professores, futuros pais, futuras mães, atuais mães, futuras avós, que terão levado consigo um pouquinho mais de conceitos que já são tão claros, mas que precisam ser repetidos em relação à promoção de saúde, prevenção de doenças e melhoria de qualidade de vida, né? Então, eu digo que aprendi muito essas 6 semanas, agradeço imensamente a Erika, Cris, Paola, pela parceria, Alejandro, aqui, muito, muito gentil, Alejandro, thank you so much for staying with us here, and you are so kind with us. Muito obrigada mesmo, agradeço a todos os professores que prepararam, eu só queria trazer isso para os estudantes, que eu acho que é bem importante, it's very important. Os professores prepararam as aulas com muito cuidado, they kept pay attention to prepare the class for you, was special, they prepare just for this, is very important. A lot of professors show it here, results not published. It's very, very important to science. Estou muito, muito feliz mesmo, e acho que com muitas ideias para o ano que vem. Espero que vocês nos encontrem. A Cris comentou um pouquinho dos próximos webinars, porque é um conhecimento que continua apesar da disciplina. Então, a disciplina a gente encerra hoje. Quem, por favor, não esquecer de preencher lá os forms que faltam, a gente vai dar aí mais 15 dias para as pessoas terminarem. Mas eu só queria agradecer muito, espero encontrá-los pessoalmente, ler o trabalho de vocês, espero assistir apresentações de vocês nos próximos anos, because the difference between us and you is just the time, it's just the time, we are older. Muito obrigada, viu?
Maravilha, gente, muito obrigada. Muitas graças pela participação de todos. Muito obrigada às minhas colegas aqui, Fabíola, que foi quem motivou esse curso desde a primeira edição. Naquele Natal de 2022, tudo começou e a gente já veio na segunda edição, realmente com um trabalho muito lindo. Paola, Eri, querida, muitíssimas gracias por todo o apoio, por toda a colaboração de verdade, não foi apoio, foi colaboração. Trabalhamos juntas todo este tempo, já há 6 meses organizando esta segunda edição, um curso que uniu 3 países em la organização e 7 países em su desarrollo. Então, estou realmente muito feliz. Muito obrigada a todos os que participaram e a gente se vê, né, nos próximos eventos. Não nos deixem, vocês vão continuar recebendo nossos convites.
Muito obrigada. Alejandro, você não quer falar algo, por favor?
Sí, ya falou.
Trabajar con ustedes para mí es inspirador, las admiro, las quiero ya después de todo este tiempo. Me parece que aprendemos a construir equipo, aprendemos a respetarnos, trabajamos desde un lugar de mucho respeto y de mucho reconocimiento y creo que es sumamente placentero y creo que eso se va a ver expresado en Buenos Aires 2025. Me parece que el respeto, la construcción en equipo, la construcción colectiva es mucho más rico que el trabajo individual y aislado.
Escutei, Paola.
No, yo solo decía que Alejandro ya había hablado y está preparando Buenos Aires para recibirnos el próximo año.
Será un enorme placer. Con Enrique, con Horacio, con Débora y mucha más gente que estamos trabajando, nos comprometemos a hacerlos sentir muy bien y a que sean muy felices en Buenos Aires el año que viene.
Muy bien.
Y de tomar buen vino, no sé qué dirá Paola sobre eso, pero para tomar muy buen vino y comer muy ricas carnes.
Eso.
Espetacular.
Muy bueno. Muy
bueno.
bueno.
¿Nadie quiere ir? ¿Nos vamos?
Nadie nos quiere. Nos vamos, sí.
Alguien ha sido esto que nos cuesta ir, ¿no?
Eso. Nos vemos pronto. Nos vemos pronto.
Tchau, tchau.
Bom dia, boa tarde e boa noite.
Tchau.
Muito obrigada.